but you can see the omega system is slightly different here. Again, 18 carbons, 2 represents the number of double bonds between carbons, and these numbers represent where those double bonds are. That's the delta system. And remember, the delta system always starts from the carboxyl end. So the carboxy end, which is this one. So if you start here, you can actually count all that and actually determine that the first bond, double bond, occurs here at carbon 9. And then the omega system, very similar, but you can see it doesn't give exactly where the bonds are, but it does tell you where the first bond is. The first bond here is at carbon number 6, but remember this one starts from the methyl end, which means when you look at this, here's the methyl end, so you're starting from here, and this is carbon 6, that's where the first double bond is, the second or third, whatever it is, it's always 3 carbons away. Very fascinating. They're both appropriate depending on whichever system you become comfortable with. And there's a reason why there's 2 different systems. It just depends on. It might have been actually 2 different countries that created the 2 different systems, and now we just have to learn both systems because it just depends on the scientist. You can choose which one you want to use, but you'll see it written as both ways. So they're both sufficient. But you do need to now know both because they both exist. Keep in mind, remember that the structure of your fatty acids can be very long or very short. So we have a short chain fatty acids, which are going to be, 2 to 6. Medium are going to be 6 to 12. Chains, and the 12 meaning 12 carbons. So 6 to 12 carbons, that would be a medium chain fatty acid. We have the long chain fatty acids, which are 14 to 20 carbons long, and the very long fatty acids, which are going to be over 22 carbons. So all of those exist in our body, and we can make all of them as long as we have our essential fatty acids. So we can make all of those short, long, medium fatty acids that your body needs, as long as we get the essential fatty acids in our body. We have enzymes to create these lipids. All right, fatty acids. So these are the essential fatty acids that we're going to talk about. There's only 2 that you need to remember. And we'll go over in another chapter later in the term a lot more detail about this. So I'm going to introduce the essential fatty acids. Apparently I needed more coffee. We're going to discuss these fatty acids, but we'll talk about them in more detail later on. So there are 2 types, omega-3 and omega-6 are kind of the general terms for these fatty acids. There's technically you could say there's a variety of omega-6 and omega-3s. There's a family, I should say, of omega-6 and omega-3s. And the omega-6 family, that one's primarily, so another term for that is linoleic. My finger doesn't work as well. But linoleic is omega-6, and I still confuse these guys, but keep in mind, you do need to know the difference. Linoleic is omega-6. Linolenic is omega-3. You can see why I get them confused all the time. I get them mixed up, but you will not get them mixed up because I'm going to remember them this term and not get them mixed up in my head. Omega-6 is linoleic. Omega-3 is linolenic. Omega-6s, we tend to have a lot of those in our diet because we can find them in vegetable oils, which we eat a lot of, at least here in the United States. So omega-6s aren't a problem, but we do have a tendency to have fewer omega-3s, and we'll talk about kind of why that might be an issue, that there can be deficiencies of omega-6 because omega-6 are not found in all foods or not as many foods as omega-6s are. Omega-3s are found in fish. We can find it in leafy greens, really dark leafy greens like kale. And then also seeds like flax seeds, chia seeds. So seeds will tend to have these omega-3s. And you can see why the American diet doesn't tend to get a lot of omega-3s, because we don't tend to eat a lot of these foods. But we need more omega-6s in generally anyway. It's usually 3 to 1. We need 3 times more omega-6s, we think, compared to omega-3s. But we need both of them. Both of them are essential to have a healthy life. And that's because they make these guys. These guys are called eicosanoids. Eicosanoids are hormone-like substances. And the eicosa is actually, it means 20. And that's where the name comes from, because these guys that the omega-3s and omega-6s are making are going to be 20, eicosanoids are 20 carbon long, 20 carbons long. That's why they're called eicosanoids. And they are inflammatory, the ones that are made from omega-6, those eicosanoids are inflammatory. The eicosanoids made from omega-3 are anti-inflammatory. So you can see that there's a difference in their function, even though they both are used to make eicosanoids. The eicosanoids made from omega-6 tend to be inflammatory, and the eicosanoids made from omega-3 are anti-inflammatory. And keep in mind, it doesn't mean omega-3s are good, and omega-6s are bad because they're inflammatory. What that means is we need inflammation sometimes. Inflammation is a way for our body to clear itself from pathogens that might be in your body, and so we need inflammation to happen. So omega-6s are just as important as omega-3s. We just tend to be out of balance. We tend to get more omega-6s and not enough omega-3s in our diet, which that is what can make it a problem. Yes.
Are eicosanoids related in any way to prostaglandins?
They are prostaglandins. So yes. And that's what we're going to talk about actually in the next slide. Not the next slide. But we are going to talk about them in relation to prostaglandin. So these eicosanoids, there's 3 types we're going to talk about. We're going to talk about prostaglandins, thromboxanes, and leukotrienes. These are all three of those are eicosanoids. They're 20 carbon long hormone-like substances that are inflammatory or anti-inflammatory. But yeah, prostaglandins is just a type of eicosanoid. Did you have a question?
No, I answered my own question. Thank you.
And these will help us maintain healthy skin, healthy nerves. So they're really important for our nervous system. The brain, about half of the lipids in the brain are actually omega-3s. So you can see how important omega-3s are in our nervous system. And here's just the picture of alpha-linolenic, so omega-3, versus linoleic, which is omega-6. You can see that they are unsaturated. So you have 3 points of unsaturation with omega-3, and you have 2 points of unsaturation for omega-6, linoleic. So that's what they look like. And I'm going to talk just briefly about these omega-3s. We actually, during the 2000s, we're doing research on, in Greenland, so indigenous population of Greenland, they were eating diets very high in fat. And by the way, in the 90s, there was this whole, let's get rid of fat from our diet thing going on. So there was like a lot of sugar and not a lot of fat in our diets because people were trying to lose weight by not eating fat, which is kind of ridiculous. I see where their thought was. But ultimately, we discovered that isn't working. We're not getting healthier by eliminating fats. And we studied these Greenland Eskimos, this indigenous population, and they were eating so much fat in their diet, and yet they didn't have the rates of cardiovascular disease that we had in the United States. So that made us realize that fat and cardiovascular disease, maybe they're linked, but omega-3s, people who tend to eat a lot of fats that have a lot of omega-3s, were having lower rates of cardiovascular events. And so we started studying them, and we started telling Americans we need to eat a lot more omega-3s. And that's because Eskimos, even though their diets were high in fat, they had very low rates of heart disease. They had lower rates of blood clot formation, lower rates of inflammation, including arthritis and asthma. They had lower rates of irregular heart rhythm, lower rates of cancer. So the scientists at the time assumed it must be the DHA, it's just a type of omega-3, docosahexaenoic acid. So, or icosapentaenoic acid. So these, I usually just call them DHA and EPA. DHA and EPA are omega-3s. So if I say DHA or EPA, they're just omega-3s. They're two types of omega-3s. These are the ones that you find high amounts at in your brain. So your central nervous system tends to have a high amount of these DHAs and EPAs. And people, or at least the Greenland, endogenous population of Greenland, were having lower rates. And so we tried to do this with Americans and give them a lot more of omega-3s. We started supplementing people with omega-3s. However, what we found is it did not decrease rates of heart disease. So there's something about the culture that you grow up in that affects your DNA, the epigenetics of your DNA. And so just because you have an endogenous population eating a lot of omega-3s and are healthier doesn't mean that you can take that same diet and give it to another culture and it's beneficial to them. So what I'm trying to say is that don't always assume because our DNA is different. We evolved slightly differently. So if you come from a different generational history, your DNA is going to be slightly different epigenetically than the DNA of someone who grew up in a different culture. So always keep that in mind. It's not necessarily, there's a lot of gray area when it comes to changing people's diets. It's not one diet that is great for every person. It's variable. Yeah.
Since salmon can be high in omega-3 fatty acids, is
Yeah.
it better, or not better, but do you receive more nutrition from eating sashimi and even
I want to say that whenever you cook lipids, it will they can be pretty stable, but whenever you cook them, whenever you add heat to them, you are going to start changing their structure. So generally, I don't want to say it's healthier, but I will say that there might be more benefit coming from raw in this particular case. But having said that, when you cook it, especially if you don't overcook it, you're probably not destroying it that much. I would say that the lipid nutrients, yes, they're going to change when they cook, but as long as you don't overcook them, I would say either way is probably beneficial. But maybe raw is a little bit better. It's really just depends on how you do it. But omega-6s and omega-3s, the reason why there's this huge difference, they both make eicosanoids. So they both make prostaglandins, thromboxanes, and leukotrienes. I feel like something's missing from the slide, but I'll go with it. So they both, and these are all the eicosanoids. Omega-6 makes them from arachidonic acid. I just see another spelling error. There should be an H, arachidonic acid. When did I make these? But arachidonic acid is an omega-6. So if you see a test question or board question asking what do we use to make arachidonic acid in the body, it's omega-6 fatty acids make arachidonic acid. And arachidonic acid makes these eicosanoids that are inflammatory and they increase blood clotting. And they're arrhythmic, which means they can affect the heart rhythm. Whereas omega-3s are anti-rhythmic, so essentially they're going to help with the heart rhythm. They'll decrease blood clotting, and they're anti-inflammatory. So just so you know, thromboxanes tend to affect blood clotting. They're all eicosanoids. Prostaglandins, thromboxanes, and leukotrienes, these are all eicosanoids. But prostaglandins tend to affect the heart a lot more, as well as be inflammatory. Thromboxanes, these really are about blood clotting. So the thromboxanes made from arachidonic acid will be enhance blood clotting, whereas the thromboxanes made from omega-3s or EPA and DHA are going to decrease blood clotting. So you can see how they are competing with each other. And that's why it's not, you don't want one or the other, you want a balance of both of them. Because you do want your blood to clot sometimes, but you don't want it to clot too much. You do want inflammation, but you don't want too much inflammation. So really, all this stuff is about balance. Everything that you're going to be doing health-wise when you're working with your clients is trying to balance them out. All right. The risk, though, Greenland Eskimos, even though they do not have high rates of heart disease, they actually do have increased rate of hemorrhagic stroke. So hemorrhage means you bleed out. And that's what's going on. So if you have too much omega-3, too much is not a good thing either, because remember, it is a blood thinner. It will decrease blood clotting. And so they have a higher risk of hemorrhage. So again, it's about balance. There's a lot of different blood thinners. So for example, aspirin acts as a blood thinner, and that's because it actually does affect these guys. So we'll talk about that later. And vitamin E acts as a blood thinner. So there's a lot of different types of blood thinners. There's a lot of things that can affect blood clotting. And these just have, are one way, these molecules can affect blood clotting too. I don't know if that answers your question, but we won't go over the pathway of how they do it, no. That's going to be beyond the scope of this class. So again, genetic component is always there when it comes to nutritional status. We can't assume that just because omega-3s, the diets for the endogenous populations of Greenland, just because they have good health with that, we can't just give Americans omega-3 supplements and hope that they have fewer rates of heart disease. Because we did that. We've actually done a lot of studies where we've given people supplements of fish oil or omega-3s. They haven't worked. We still have high rates of heart disease. So again, a lot of the information when it comes to diet can be gene specific. So it just depends on the culture, the epigenetics of your genes, and that's going to play a big role in as to whether or not a certain diet change might be beneficial or harmful. But we do know if you do not get omega-6, the main deficiencies are poor growth, scaly skin, because you really need omega-6s for healthy skin, for omega-3s as well. Deficiency of omega-3 is more harmful because you have neurological and visual abnormalities. Again, remember, 50% of your brain is DHA. 50% of your brain tissue is omega-3. It's actually DHA. So it's going to have definitely neurological issues if you don't have enough of that in your diet. And this is where you find them. You don't have to memorize this, but it is probably good to know that most of your omega-6s, which again are your linoleic acid, these are found in a lot of your oils. Our plant oils tend to have a lot of those, whereas omega-3s tend to be less found in our diet, but we do find them in a lot of fishy foods, but also in a lot of seeds. So where's the seeds? Okay, seeds aren't on here. But we have it in certain seeds like chia seeds and flax seeds and other things as well. So there are good ways to get omega-3s in your diet. All right, so that was just an introduction for your essential fatty acids. Again, we're going to talk more about their metabolism and the eicosanoids later in the term. Actually, I think in a couple weeks. But now we want to focus on what we do with fatty acids. So we know what fatty acids are now. We know their nomenclature. We know that they can be saturated or unsaturated. But now we're going to make stuff with those fatty acids, and some of those products are going to be triglycerides. So triglycerides are 3 fatty acids, that's where the tri comes from, attached to a glycerol backbone. That's where the glyceride comes from. So essentially you have, what is a glycerol? A glycerol is 3 carbons, 1, 2, 3. That's a glycerol. It's a little more complex with that, but it's basically a 3-carbon molecule. And each one of these carbons is going to be connected to a fatty acid. That's a triglyceride. So a 3-carbon molecule, which is a glycerol, this is a glycerol right here, and 3 fatty acids are going to be, or 1 fatty acid attached to each one of those carbons. So if you just have a diglyceride, then you remove one of these fatty acids, and now you have a diglyceride. If you remove another fatty acid, now you have a monoglyceride. So 1 is a monoglyceride, 2 is a diglyceride. And another way to put it is glycerol. Actually, it'll be in my slides. I'll show you later. All right, so again, triglycerides, here's a better visual of it. You have the glycerol backbone here, 3 carbons there. You have the fatty acid chains, 1, 2, 3. You can see the long chain is that long hydrocarbon. So these 2 happen to be saturated, and this one happens to be unsaturated down here. So you can have saturated or unsaturated in your triglyceride. They can be all unsaturated or all saturated or a mix of both. And this is essentially what all your oils and butter, all of the fat that all the lipids that you eat are primarily in the form of triglycerides. So the butter, the pizza, the hamburger, all that good stuff, 99% of the lipids in that are in the form of triglycerides. And that's also what you store in your body. So your adipocytes, your fat cells, are storing triglycerides. So 95% of the fat in the body and the fat that we consume is in the form of triglycerides. Oh, and I mentioned it's an ester bond. Do you remember that? Functional groups. This is an ester bond, and it's made with a carboxyl end. So the carboxyl end of the fatty acid is what attaches to that glycerol backbone. And it's called an ester bond. All right, so I'm just introducing the structures of the lipids that you need to know. So I've introduced the structure of fatty acids. I've introduced the structure of triglycerides. Now I'm going to introduce the structure and general function of sterols. So sterols have a very unique ring structure. It's actually 4 rings, 4 connecting rings of carbons and hydrogen. And carbons and hydrogen, that's why it's hydrophobic. If there's a lot of carbons in something, it's going to make it more hydrophobic. And remember, that's when something can be labeled a lipid, when it's hydrophobic, when it doesn't like water, and it's got a lot of carbons attached to it, then it's a lipid. And cholesterol is a lipid for that reason. Cholesterol is the most common sterol in the body. By the way, it is not found in plants. Cholesterol is only found in animals. When I taught a nutrition class, one of the questions would be, where do we find cholesterol in the diet? And students would Google the answer, and plants would come up. That's why you can't Google nutrition questions. You'll see this when we get later into vitamins and minerals. If you start Googling vitamins and minerals, a lot of weird information will come up. So you definitely want to make sure you're looking at the textbook, not Google when you get to some of this stuff. Because cholesterol is an animal product, and our bodies make it, so it is not essential. We do not need it from the diet, but we do eat it because any of you that eat animals are going to get cholesterol in the diet. It's not found in plants. Plants do have sterols, just not cholesterol. Cholesterol is specific to animals. And it plays a lot of really important roles in the body. We need it for the cell membrane structure. Most of the lipids in your cell membranes are unsaturated, which remember that gives them a kink, and so they tend to be more flowy. And if you think about what a cell membrane does, you want it to be a little bit more rigid. And that's what cholesterol will do. It'll actually sit in the cell membrane and help stabilize cell membranes. So it's important for stabilizing cell membranes. It's important for creating a few important compounds in the body. I mentioned a couple yesterday. Do you remember any of them? Testosterone and estrogen, the sex hormones are definitely made from cholesterol. There's an important, what was it? Progesterone is made from cholesterol. There's another really important vitamin that's made from cholesterol. Vitamin D. So we actually have a cholesterol form, it's a dehydrocholesterol sitting in our skin, and when the sun hits our skin, it actually starts making vitamin D. So we need cholesterol to make vitamin D. It also is needed to make bile salts. So important for a lot of important things. But it is not required in the diet because our body can make cholesterol. And we'll talk about how we make cholesterol probably next week. But this is what it looks like. Again, it's a 4-ring structure. Every time you see a point here, remember there's carbons. There's carbons at every one of these points here. These are all carbons. So all of these carbons make this structure a lipid because all these carbons are not charged and they don't want to interact with water. So that's what makes sterol a lipid. And again, cholesterol is a type of sterol, and it's important to make a variety of compounds. Vitamin D, testosterone, estrogen, progesterone, bile salts, and it will stabilize cell membranes. All right. So that, you should be able to now tell the difference between a triglyceride and a cholesterol, or sterol. Now we're going to look at the structure of phospholipids. So phospholipids contain a phosphate. So phospholipids have a glycerol backbone again. So again, we're going to start off with a glycerol backbone, which is 3 carbons. But phospholipids only have 2 fatty acids attached to the glycerol. And they have, on the third one, they have a phosphate group. So that's what makes phospholipids different from triglycerides. They only have 2 fatty acids, and they have a phosphate group. And this is going to make phospholipids very hydrophilic, at least the head portion. So this portion right here, the glycerol and the phosphate group, this is what's hydrophilic. But the fatty acid tail is very much hydrophobic. So these 2 fatty acids hanging off of the carbons are very much hydrophobic. And remember, cell membranes have a phospholipid bilayer. Let's look at that. I'm going to show you this real quick, and I'll go back to that slide. So remember the bilayer, you have up at the top here, this is the hydrophilic portion because it's interacting with the water. So the hydrophilic portion is on the bottom and the top. This is where you have, this is the glycerol backbone, and the phosphate group are going to be the head groups of the lipid. And then you can see in here, these are the fatty acid tails in the middle here. So cell membranes are always lipid membranes, bilayer. So there's 2 layers of phospholipids, one at the top, one at the bottom, and they both come together to form the cell membrane. So here it is, glycerol backbone, phosphate group, 2 fatty acids, and we can make a phospholipid. The head group is polar. In other words, remember it's hydrophilic. Try not to hydrophilic. And this tail portion is hydrophobic. Another way of putting that, remember this, another way of saying hydrophilic is polar. Hydrophilic and polar, same thing. Talking about the same thing. Hydrophobic and nonpolar, talking about the same thing. So hydrophobic and nonpolar go together. Hydrophilic and polar go together. Remember, water is polar. So hydrophilic is water loving. So they're important. So what are their functions? They're important in cell membranes. They make up the cell membranes. We're going to talk later about lipoproteins, but those are going to be your HDLs and your LDLs. your lipoproteins. We'll talk about those. They can also act as emulsifiers. So you can find them associated with bile salts, which are also made from cholesterol, which are all made from cholesterol. Or I shouldn't say all, but a lot of the bile salts are made from cholesterol, but they do have emulsification. Phospholipids can have emulsification purposes. If you're a cook or a chef, you know emulsification is basically when you take salad dressing, you add olive oil and whatever vinegar you're using and shake it up. That's emulsification. So we'll talk more about that later. And then structural support, phospholipids are going to provide a lot of structural support for the cell as well as cell signaling. We're going to get deeper into these functions. We're really just introducing them now. So again, this is its function. Its main function is to create these lipid membranes. And this is what I'm saying with emulsification. So you can actually, when we digest food, this is really what happens as well, but you have these phospholipids that come together, and inside of here, this is a fat droplet. So they actually surround the fat droplets, and that way it can be in these aqueous watery solutions, and the fat droplets can hide from the water. So they're hiding from the water when you emulsify them. Here's another more biochemical picture of what a phospholipid should look like. And so you see here we have a saturated fatty acid and we have an unsaturated fatty acid. Again, they could be both saturated or both unsaturated. It just depends on the molecule. Here's the glycerol backbone. Here's a different way of looking at that. Notice it's not cartoonish. Like I just wrote it as 3 carbons. But this is showing the 3 carbons, 1, 2, 3. They're showing it in a different way here. So, but you do have a carbon here, a carbon here, and a carbon here. So it's still a 3-carbon molecule. And then you also see on this 3rd carbon, the one that does not have a fatty acid, is where you have this phosphate group here. But again, it's the same thing, and this is what it looks like. So you should be able to recognize the structure differences between phospholipids, or sterols, and triglycerides.
Excuse me, can you identify the 3 carbons one more time, please?
Yeah, I can try to on this picture. Oh, here we go. So this in pink here is the glycerol group in the pink color, and the carbons are actually here. Gotta change it. Here's 1 carbon, here's the other carbon, and here's the other carbon. Did you guys see that? Here's 1 carbon, another, and the other. Yeah, it's just, I forget I can do this. I love using an iPad in class. It's so much easier. All right, I have this slide in here, so I'll abbreviate it. You can kind of ignore it if you want to, but this is just showing there's lots of different lipids. We're going to talk later about the differences between phospholipids and glycolipids because we have a lot of membrane lipids, and so some of them are not going to be phospholipids. Most of them are, though. So this was just, I was going to try to introduce you guys to all of these now, but I think I'm going to save this for later because I don't want us to get too much in the weeds with this slide. Because we'll talk about these different lipids as we move forward in the term. So I'm just going to get this slide. Yeah, cool with that. Good. And let's just focus on digestion. I want to keep this simple. I'm going to, I know I have those review lecture videos that are posted, but I really want to focus you in class and what are some of the things that I see students missing on the exams. So that's something that you get from class that you won't see on the lecture review videos. Because there were a lot of things that students missed last term, and I realized I need to make sure I clarify that with you guys this term. Because these are important pieces of information. So we're gonna digest all of them today, starting with triglycerides. If we have time. Yeah, we have time. So we're not going to get, by the way, probably to lecture 2. We might be able to start it. But don't worry, when we created this biochemistry 2, we're gonna have a lot of spillover the first couple of weeks. So don't fret, there's still plenty of time to get through the material, but I don't want to go too fast. All right, triglyceride digestion. This one, when you are digesting triglycerides, it is always a lipase. Lipase. We don't use lipases to break down the other lipids. That was the thing that I saw students missing a lot on the exams. They saw lipase and they think break down lipids. Technically, these lipases are breaking down triglycerides. Not cholesterol esters, not phospholipids. Lipases break down triglycerides. And we have a lot of lipases. And whenever I talk about a lipase, know that I'm talking about we're breaking down a triglyceride. The first lipase I'm introducing you to is lingual lipase. So your textbook it says within it that triglyceride digestion starts in the stomach. I disagree with your textbook because the enzyme, the very first lipid or triglyceride enzyme that we have, comes out in the mouth. It's lingual lipase, and it comes from the salivary gland. So technically, in my opinion, triglyceride breakdown begins in the mouth because that's where we have the first lipid enzyme. But your textbook will say it starts in the stomach. So there's one quiz question that you might run into this week that asks you, based on the textbook, where does lipid or triglyceride digestion begin? The answer is going to be the stomach. I will not ask you a question like that on the exam because your book disagrees with me. So I'm just going to ignore a question like that on the exam, but you will see it on your quizzes this week. So if you do see that one, where does lipid or triglyceride digestion begin, you're going to have to say the stomach, even though I'm telling you, I think it's the mouth. The stomach does have a lipase as well, though. And I will say that a lot more breakdown is going to occur in the stomach than the mouth. And that's why the textbook said that the stomach is the first area where you really get breakdown of lipids or triglycerides. So in the stomach, you have a different lipase that can handle the pH 2, because remember it's really acidic. So the first lipase is lingual lipase, comes out from the salivary glands in the mouth. The second lipase is gastric lipase. This one is secreted in the stomach. And what they both do, they're taking off the fatty acids. So when you have this glycerol over here, I'm just going to make a glycerol. So when you have a triglyceride, and it's got these 3 fatty acids attached, what the lipases are doing are breaking the ester bond. So they're taking off the fatty acids from the ester bond. They're breaking the ester bond to release the fatty acid. That's what lipases do to triglycerides. And when they do this, they usually can only attack from the sides. They really can't reach the one in the middle. So we don't usually get full breakdown of triglycerides. And I'm mentioning that now because you'll see that in the next slides coming up. So when we break down with lipases, we release the fatty acids, and we tend to end up, as we release them, for example, when we release one fatty acid, what does this 1 and 2 mean? Remember, there's 3 carbons in our triglyceride here. So if we have something called a 1,2 triglyceride, it means that the fatty acid is here still and the fatty acid is here still. But carbon number 3, because this is carbon 1, this is carbon 2, this is carbon 3. So if you get rid of this fatty acid from this one, if this one's gone, now you just have a 1,2 diacylglycerol. And that's where this name comes from. When you release one of these fatty acids, you only have 1,2 diacylglycerol. Okay, one more thing I need to tell you guys so that you stay on the same scientific language page that I'm on. there's two, so triglyceride, I gotta get better at this. Triglyceride is the same thing as triacyl glycerol. Do not confuse them, they're the same thing. Triglyceride is just an abbreviated way of saying triacyl glycerol. Acyl is just referring to the fatty acids. So whenever you see acyl, it's referring to the fatty acids. And we tend, scientists, me, whoever, we tend to abbreviate it, and I get rid of the acyl because it's already assumed that it's there, and I just say triglyceride. But technically, if I'm saying it properly, I would say triacyl glycerol. That would be the more correct term. But either one is acceptable, so I just kind of go with it. So that's why it says 1,2-diacyl glycerol. There's two fatty acids attached, but this is just a diglyceride. There's two fatty acids, it makes it a diglyceride instead of a triglyceride. Yeah.
So you were saying when it says the 1,2-diglycerol right there, the 1 and 2 stay free.
Yeah, in this case, the third fatty acid was removed. The carbon is still there, so you just have the 3 carbons, carbon, 1, 2, 3, fatty acid 1, fatty acid 2, but the fatty acid on number 3 is missing. Yeah, so that's now, this molecule would be 1,2-diacyl glycerol. If you see triacyl glycerol, you don't need numbers because that means that all of the carbons are attached to a fatty acid. So you will only see the 1 and 2 or the 2. We'll get to it later. I won't try to get into it now. But yeah, I'll describe this even more in a minute when we finish with triglyceride digestion. Just so you know, short and medium chain are going to be more easily metabolized by the body than the longer chain. The longer the chain, is going to be harder for the lipases to attack them. They can still do it, it's just going to take longer. So the medium chain, the shorter chain, the shorter the fatty acid, the easier it's going to be broken down. So emulsification starts occurring as well because of peristalsis. So I can't remember how much physiology you guys have had, but peristalsis is just the movement. I always show with both hands. It's just basically, it's involuntary, but it's the muscles of your GI tract that help move and propel the food through your GI tract. That's what peristalsis means. It's just the smooth muscle that's contracting, helping move the food through your GI tract. And that's also going to, that movement of peristalsis is also going to help start breaking up the fat. So it's a mechanical digestion that's also occurring with peristalsis. And fat does help contribute to satiety, so it's a good way to have a balanced diet. Fat is really important, and it does contribute to satiety because fat in the stomach will delay emptying, so it'll help you feel full longer than eating like straight carbohydrates. Yeah.
What would like an example of an undigestible fat be?
Oh, this is just saying what this means is that as the fat's not digested fully yet.
Gotcha.
And so it takes a while in the stomach. It'll sit in the stomach for a while, not being fully digested.
Gotcha.
Yeah. Okay. Still doing triglyceride digestion. We have only made it through the stomach. Now we have to get to the intestine where there's another lipase. What do lipases break down? Which lipid do lipases break down? Triglycerides. Good, we've learned something today. All right, we have another lipase, though, that comes out in the intestine, pancreatic lipase. So lingual lipase, gastric lipase, and now pancreatic lipase. Yeah?
I just have a quick question. So are triglycerides considered fatty acids?
So triglycerides, I think it's a good question, because triglycerides, remember, there's that glycerol group, which is 3 carbon, and then you have 3 fatty acids attached. And so these chains can be long, short, it doesn't matter. It actually has nothing to do with so the triglyceride itself can have one chain that's short, like a short chain, one chain that's long and the other chain that's medium. What the fatty acid is. So the triglyceride will always just be called a triglyceride, but the fatty acid chains can be anywhere from 4 carbons long to 10 carbons long to 20 carbons long. Yep. So this pancreatic lipase is going to come out in the small intestine and it's going to finish digestion of triglycerides. So what's going to happen with these triglycerides, so we have, again, the 3 carbon molecule, triglyceride. Remember I told you the lipases will work on the ones, so here's carbon number 1, carbon number 2, carbon number 3. The lipases will work on carbon number 1 and carbon number 3, but they have a really hard time getting to carbon number 2. And so we tend to absorb, it's called, the actual one that we absorb is called 2-monoacyl glycerol. That's what we absorb, 2-monoacyl glycerol, because lipases tend to not be able to break down this final portion right here. So, and you'll see this, what digestion ultimately ends up with is this primarily. So I do have this because sometimes we can absorb the diacylglycerols, but I'm going to mark that out because I'd rather you remember this one. It's really, the main result is going to be monoglycerols and fatty acids. You might have some free glycerols, but even this one is rare. So I might even change this where I actually have it where these two are the main components. These are the main things that your body is able to make. The reason why you tend to not have free glycerol is because your lipases tend to not be able to get to that last fatty acid. Having said that, there are you will have some free glycerol. Those lipases, some of them will attack that monoacyl glycerol, and so you can actually, if that's attacked as well, then you can end up with free glycerol. It just doesn't happen as much. The main product after digestion is over are going to be these two things: monoacylglycerols and free fatty acids. And that's what's going to be absorbed from triglycerides. Yeah. So I'm focusing you guys on what I really want you to know. Yeah. So focus on if I'm saying this is what I want you to know, then that's what I want you to know for the exam. And for the boards is probably what they're going to. So I was going to be more broad, but I will say when I was looking at some of the stuff, the boards might want you to be more specific. I want you to know there's gray area, which is why I tell you the other two can be made as well and be absorbed. But if the boards is going to be focusing you guys on what's black and white, these two products are really black and white. These are the ones that you really do absorb. So focus on these two. Monoacylglycerides and free fatty acids is primarily what you're going to absorb. But don't get some of the others too. All right, so here's just kind of showing you a picture. So you start with a triacylglycerol, and when the lipases start their work, they've taken off this fatty acid here, and when they do that, you end up with 1,2-acylglycerol and a free fatty acid. And then when they keep working, do I have another one? I don't have another one. So when they keep working and they take off this fatty acid, which they will do, then you're going to end up with just 2-monoacylglycerol. Does that make sense? Yeah.
So when we make it to the pancreas, is that third carbon that we lose in the gastric, is it gone? It's gone for good, right?
So I would say that with the gastric lipase, they're all doing the same thing. And so what's happening in the stomach is it's just doing its best to try to pull as many fatty acids off as it can before it gets into the small intestine. So it doesn't mean, like, it's primarily, but it's not going to be full digestion. So it could actually, though, attack both carbons, and the gastric lipase could make the 2-monoacylglycerol too, which means its digestion will be fully completed by the time it gets to the intestine. But generally speaking, the gastric lipase, because it's not there very long and the lipases don't have a really good opportunity to attack all the different triglycerides, you don't have complete digestion in the stomach. But it could potentially take off both fatty acids and end up with monoacylglycerol.
And that's the end goal every time, 2 monos, where we're trying to get to whether you.
The main end goal, is the monoacylglycerol and free fatty acids. And all the lipases do the same thing. They're just taking off the fatty acids. And they just can't seem to be able to make it to that middle fatty acid. All right, so that was triglycerides. Digested, the enzyme that digests them is a lipase. And again, there's lots of different lipases. We'll talk about more in later weeks. There's hormone-sensitive lipase. There's lipid protein lipase. But all lipases do are attack the fatty acids and pull them off of the glycerol. That's what they do. Okay, now we need a break. And then we're going to talk about cholesterol and phospholipid digestion, and I'm going to set up quickly real quick.
You ever seen him sing
It doesn't help. It doesn't help.
with Michael? Just emphasizes the.
It makes you want to actually pay attention. Yeah.
That shit.
You okay, Jack?
Yeah, what?
You're falling asleep.
No, recommend it. Yeah, like you gotta study.
God, I think we lied about this. I think it's single text, a call. You didn't text me at all. I was hooked up to your mom. I was gonna say you should really turn around and think about it. You didn't text me at all.
It sounded better. I was watching. Hey, I'm the clutch dog, right? I'm the clutch dog, right? I'm sit. Am I the clutch dog? Do I clutch up? Always. All right. All right.
So,
Clutch dog.
I mean, I just was saying. Yeah. But I would say, yeah. So I won't worry about it. Sorry, because I forget. I usually do it right before yeah, break time, and I might do that, but I don't have. I don't want to set the time.
That's like I know I do. No, I literally don't. It's embarrassing.
So last term,
Yeah, exactly. A jar of like
Whether they're having this course.
Because I completely
I just wanted to check. Dude, I think, sorry, I don't.
I got a sign on
I know. It's just like,
they're weird.
I just wanted to check it.
I'm not talking about
I just wanted to check it. So when you
I'll send it to you.
like your audio system will be PowerPoint, but it's like it's just then you
just go through it makes it to me like I look that way. That's why I think I'm like learning space of how we kind of do things. So
I just wanted to check.
Hey, real quick, I just want to do a sketch just to make sure I get this down. So we got the three life phases we talked about, lingual, gastric, and pancreas. So you have your three carbon chain, simple, and then you have your fatty acids, right? That during from mouth to gastric, they'll attack these two. Because it's protected, correct? So fatty acids attack these two. And then once you move in, this doesn't get down like it's in there at the end. That's when this one will kick in. It removes this one out, right? So this one becomes completely gone. This one becomes completely gone. This one can't be attacked. This one has to be gone, right? Okay, perfect. Like, I don't want to complicate that. That's pretty much
that
perfect.
is actually it. So what we absorb is are 2 monoacyl withdrawals and 3 fatty acids.
And free fatty acids, which is what you boxed in. I put that in red on my notes. Okay. So just remember 2 and then fatty acids is what we got. And that's it. Okay, perfect. I'm kind of thinking about like this is the quarterback and football, these are the linebackers. They're protecting these guys are blitzing. One gets to them on first down. That's what we got. You perfect.
You know what? We're going to talk about that a lot. I mean, I will tell you, like, it's basically emulsification, if you want to put it really simple, it's where you take a large fat droplet and you break it up into small fat droplets. And so basically, yeah, it's because when you take a large fat droplet and when you emulsify it, so it's that kind of like, yeah, it's like, you think of salad dressing if you shake it up. But if the short way of thinking, like, you take a large fat droplet and like, yeah, so we're surrounded by the phospholipids that surround by the fat inside the middle. Yeah. And we'll see what pictures of it. No, like that's your question.
Okay, so I got to think. Lotus? I love it.
Did you laugh? In a I hope so. Without fail. Yes, yes, yes. The PM. Nice.
So have I talked to you guys about Jeff Bezos yet?
What?
Jeff Bezos. You guys know who he is, right? Amazon man. Pure intention. 1%? Oh yeah, he's definitely one of those. 0.1%.
I'm listening.
All right, guys. So, how do you want to be? Just a little bit of my worldview. Jeff Bezos is really interesting, as a lot of variety of people are out there. He made this. I watched an interview with him, and he talked about what propelled his career or focus. He started out with one very pure intention. He wanted to make, and he was hyper focused on this idea. He wanted to make a company with the absolute best customer service. He didn't care about money. He didn't care about anything else. He was focused on how can I create a company with the best customer service. This is before Amazon became Amazon. And the reason I mention this is because in my worldview, this is consciousness. It is, you are creating your reality with the thoughts that you cultivate and the ideas that you're cultivating now. And so if you start with a very pure intention of the type of population you want to work with, whatever it is that you want to get from your chiropractic career, like start doing it now. Start building that momentum towards that by putting those ideas, surrounding yourself with those thoughts. And I'm telling you, in some years later, 5, 10 years, you're going to see those really pure, beautiful intentions that could make you a billionaire, just like they made him a billionaire. Because when you have those really pure intentions, then stuff comes. And that's what I want you guys to start thinking about when you're working on figuring out what you want to do with your chiropractic career. Start nailing down an idea that you can start waking up with every morning and going to bed with every night. And I promise you, something's gonna happen when you do that. You're going to be filled with energy. And it's gonna help you get through these classes too. I know when I started out with my intention of being of service, that helped me. So when I come into work, I am tired some days, and I don't like that I am. And so I remind myself how much energy I have. And I also remind myself how much I love hanging out with you guys and talking to you guys. And it really does, like, it's fed me. So think about that as you're thinking about your career and getting through this graduate school, which I call graduate school my dark days. I don't want you guys to have to do that. I didn't really start out with pure intentions. I didn't really know what I was doing in graduate school. But I'm just putting that out there as I was waiting for people to come back in. Because we are going to get focused again on digestion. So we have already digested triglycerides. We're about to absorb the triglyceride breakdown products. But first, the body also needs to digest cholesterol and phospholipids because they are a part of your diet as well. And these are going to be different enzymes. Lipases do not break down cholesterol esters and phospholipids. So again, we do get cholesterol in the diet if you're eating animal products, as well as phospholipids, so they're just part of cell membrane. So a lot of the foods, plants that you eat are going to have phospholipids. So in order to break down cholesterol, most cholesterol that you eat in your diet is esterified. What does that mean? It means that cholesterol, that four-ring structure, okay, I'm just going to draw 4 balls because we know that I'm not going to be able to draw cholesterol. But it is a 4-ring structure. And when we want to store cholesterol, and a lot of the cholesterol that you consume is esterified, meaning there's a fatty acid attached to this guy. And when you put a fatty acid on it, in order to absorb cholesterol from the diet, you have to break off that fatty acid. And so what's going to do that, and that's how you break down cholesterol, so you basically just have to take off this fatty acid, and what does that is cholesterol esterase. That's the enzyme that will take the fatty acid off of the cholesterol. You still have a cholesterol, so what you're going to absorb then is a free cholesterol and a free fatty acid. That's it for cholesterol. And this happens in the intestine, in the small intestine. Phospholipids, remember they have fatty acids attached to them. So you have a glycerol, so 3-carbon glycerol, and 2 fatty acids attached to the phospholipid. And so in this case, you're going to have to take off the fatty acid. And the enzyme that's going to do that for phospholipid is phospholipid A2. Phospholipid A2 is the enzyme that breaks down phospholipids. Not a lipase, although it does say phospholipase. But in this case, there's always a phospho there. So don't get it confused. It's a phospholipase that breaks down the phospholipids. And what they make, when you break this down, and I'll show you a picture in a second, try to draw it out for you, is you make a lysophospholipid and you get a free fatty acid. And that looks like this. So here's the phospholipase A2, so that's the enzyme, and it's going to break off this. So this is the fatty acid. It doesn't look like it because they abbreviated the fatty acid. Instead of drawing out all of the carbons, they just put R. So an R in chemistry just represents a functional group. In this case, the functional group is a fatty acid. And so when you take off this fatty acid, this phospholipase A2, oh wait, actually that's not it. Sorry, give me a second. Oh yeah, it is. Sorry, it is. It is. Here's the other fatty acid. So remember, phospholipids have 2 fatty acids. So here's one fatty acid. Here's the other fatty acid. And phospholipids also have a phosphate group. So here's the phosphate group. So you can see it's written a little differently than you're used to seeing it. But when you take off the fatty acid, and you only take off one of the fatty acids, so you end up with a free fatty acid, which is this guy here, and it's called a lysophospholipid. It is a phospholipid with just only one fatty acid still attached. and it's called a lysophospholipid. So you end up with a lysophospholipid and a fatty acid, and the enzyme that does it is a phospholipase, A2. And that's it for phospholipids. So hopefully I've simplified this a little bit better than I did last term. And you guys won't be confused about what breaks down triglycerides versus cholesterol esters versus phospholipids. But we haven't quite finished their digestion because what we need to do is more emulsification. So we can't, absorbing the fatty acids and getting the enzymes to properly break down the fat requires bile. You need bile to break down, to fully digest lipids. And what bile is going to do is, let's see, I kind of want to talk about bile now. All right, I don't have it here. Okay, maybe I don't have it. So what does bile do? So bile, first of all, is released from the gallbladder. What makes bile is the liver. So the liver makes bile. What do we make most of the bile salts from? What molecule? Cholesterol. So we actually make a lot of the bile salts from cholesterol. Another reason why cholesterol is really important in the body. But bile is made in the liver, and then the liver will put it out into the gallbladder to store it. I do have that written somewhere. I hope I do. Because that's definitely something important. So bile is made by the liver, it's put into the gallbladder, and essentially what bile does is take really large fat droplets, and it helps break them down into small fat droplets. Which makes sense because if you have this big ball of fat, how are enzymes going to be able to break down the lipid? And remember, your body is aqueous, and so the lipids tend to stick together and make these really big fat droplets. And so you need to break this down into smaller fat droplets. And that's what bile is going to do. So bile allows emulsification to happen, because it's bile salts. So they're going to actually have hydrophilic and hydrophobic portions. And so they are going to be able to help break down large fat droplets into small fat droplets. And now the enzymes can work on them better. So bile is a very important component of breaking down lipids. We don't need it for carbohydrates or protein. We only need bile for lipids, because we need to make them, because your body is mostly an aqueous environment, you need bile to help protect the lipids and allow the enzymes to get to them a little more easily, because you can break them down into smaller fat droplets. Here we go. So that's what's going to happen when you break these things down, and this is what your body's going to absorb. These are the major absorbed products. So you guys can tell me where we get them from. Where is the monoacylglycerol? What lipid creates the monoacylglycerol? Triglycerides. So we start with triglycerides. They're broken down into monoacylglycerols and free fatty acids. Where do we get lysophospholipid from? From phospholipids. So when we break down phospholipids, we end up with lysophospholipid and free fatty acids. And when we break down cholesterol esters, we end up with free cholesterol and free fatty acids. So these are the major components that are going to be absorbed from your diet when it comes to lipids. And we require bile in order to get to these final components. The bile is actually going to come together to form micelles. So this is a micelle, and it's made up of bile salts. So these molecules here, these are bile salts that are protecting the lipids, which are in the middle. So the lipids are all in here. And these portions out here are going to help protect that. So the micelle is going to be made in the intestinal tract. And that's actually going to be how you absorb fats. I'll talk about that in a second. So micelles can interact with the brush border. The brush border is your intestinal cells. So, all right, I'll just do it. Here is your lumen. You've just digested something, and here's your intestinal lumen, and so you have all of this digested lipids in your lumen. So what's going to happen here is these lipids are going to be formed into micelles, and they're going to interact with the brush border. The brush border is right here. It's on the sides of the intestinal lumen. They're essentially your intestinal cells. I really do wish I had my pen now. But these micelles are going to interact with the brush border here, and that's how you're going to be able to absorb those lipids and get them into the body. So, and that's what, I see what I did there. So micelles interact with the brush border to allow diffusion of lipids into the enterocyte. So the lipids, when the micelles interact with the intestinal cells, and by the way, enterocyte, what does that mean? Entero means intestine. Entero means intestine. Site means cell. So an enterocyte is an intestinal cell. Sometimes we call these, instead of an enterocyte, we call them absorptive cells. Sometimes I just call it an intestinal cell. Either way, enterocyte, intestinal cell, absorptive cell, these are all the same cells that I'm talking about. And they're what make up the brush border. So your enterocytes are what's breaking up this brush border. It's just intestinal cells. That's what the enterocyte is. And so the micelles will interact with the brush border, and that's when the lipids can simply diffuse into the enterocyte, into the intestinal cell. So fatty acids and monoacylglycerols do have other transport systems, but honestly, I'm not going to make you guys memorize them because the micelles are the really important component. But there are other absorptive systems. There's actually a fatty acid transport. Cholesterol has its own transport. It's Niemann-Pick C1. It's associated with a lot of diseases, so you might see Niemann-Pick 1 later in your graduate career here. But for now, for this class, don't worry about memorizing it. I do want you to know, though, that there are other opportunities for fats and lipids to be absorbed. But the main way that they're going to be absorbed, all your lipids, the primary way is micelles. And one other thing that I do want to point out, you definitely want to know this, medium and short chain fatty acids. Medium and short chain fatty acids do not need to use micelles. No need for micelles. They're short. They can actually diffuse in without the need for micelles. And so medium and short chain fatty acids, we'll talk about it in the metabolism. You'll see they're treated differently metabolically than long chain fatty acids. And so the bile salts, they're not waste products. They can actually be reabsorbed. We'll talk about this. You'll hear this later. Enterohepatic circulation. Entero, again, means what? Intestine. Hepatic means what? Liver. And so essentially the bile salts can be reabsorbed from the intestine and go back to the liver, so they can actually be reused and recycled. So your liver can actually maintain some of those bile salts. But a lot of the bile will also be excreted in the feces. It's what gives your feces sometimes that really dark, sometimes that greenish color. But the bile, some of it will be excreted with the feces, but some of it will be reabsorbed and sent back to the intestine, and that's called, or sent back to the liver, and that's called enterohepatic circulation. So with absorption, again, it is passive absorption. Your lipids do not technically have to have a transporter. They're lipids, and they're interacting with a lipid membrane. And so they tend to be able to just be absorbed simply by diffusion into the enterocyte. If the carbon length of the fatty acid is over 12 carbons, so it's a longer chain fatty acid, it's a short chain fatty acid, it's less than 12. If it's less than 12, if it's a short to medium chain, it will just simply go into the portal circulation. So portal system is just the circulatory system that goes from the intestine to the liver. And so medium short chain fatty acids can actually go straight into the blood and then to the liver. Longer chain fatty acids have to do a different system. So they're going to be carried by chylomicrons from the intestinal cell, and they're going to go into the lymph. Not the circulatory system, they're going to go into the lymphatic system. So short and medium chain fatty acids can go straight into the blood, to the liver, that's the portal vein circulation, but longer chain fatty acids have to be incorporated in something called chylomicrons, and I'm going to tell you what those are in a second here, and they're going to be absorbed not into the circulatory system from the intestinal tract, it's going to be absorbed into the lymphatic system from the circulatory tract, or from the intestinal tract. And again, leftover bile can be reabsorbed and returned to the liver. So chylomicrons, what are chylomicrons? This is a really long picture here. But chylomicrons are a type of lipoprotein. So this is a lipoprotein. Lipoproteins are how fat is, or lipids, I should say, are transported in the body. Remember, lipids don't like water. So if it's a long or big lipid, it needs a carrier. It needs something that's going to be able to transport them to protect them from the water. They don't like water. They need protection. And so this is protection from the aqueous portions of the body. And so in the blood, your lipids are carried by lipoproteins. And the very first lipoprotein I'm introducing you guys to is the chylomicron. So chylomicrons are a lipoprotein that carry digested fat. That's the definition of a chylomicron. It is a lipoprotein that carries digested fat. And so chylomicrons are made in the intestine. It's the only one made in the intestine. Okay, there's a little gray area there. But generally speaking, it's the primary one made in the intestine. All the others will be made by the liver. There are other lipoproteins. We have LDLs and HDLs. You've probably heard of those. Talked about them before. But chylomicrons are a lipoprotein made in the intestine from digested fat. Digested lipids. And remember, most lipids are going to be triglycerides because your diet is primarily triglycerides. 95% of the lipids that you consume in your diet are in the form of triglycerides. So that's why chylomicrons are primarily carrying triglycerides. Okay, so your body worked really hard to break down triglycerides, cholesterol esters, and phospholipids. Guess what happens when you get them into the enterocyte? You just rebuild the lipids. So inside the enterocyte, the intestinal cell, as soon as it's absorbed, you're actually going to remake triglycerides, phospholipids, and the cholesterol esters. You're going to resynthesize them. And that's going to be in the endoplasmic reticulum of the enterocyte. But the LCFA, LC just stands for long chain. FA is fatty acid. FA is always fatty acid. LC just means long chain. Medium chain would be MC. Small chain would be SC. So if you see LCFA is long chain fatty acid, MCFA would be medium chain fatty acid, SCFA would be short chain fatty acid. But you're just going to remake them. What remakes them? This guy is called acyl transferases. Remember I told you that the fatty acid, you can also refer to the fatty acid as an acyl. Acyl. That's just a fatty acid. Transferase is an enzyme. So acyl transferases are what puts the fatty acid back onto the glycerol backbone. So you have the glycerol backbone again. Acyl transferases are what's going to make the ester bond and put the fatty acids back on. And they're all called acyl transferases. Every time you put a fatty acid onto a lipid or onto anything, it's going to be done by an enzyme called an acyl transferase. So acyl transferase is just a general term used to, it's a name for an enzyme that puts a fatty acid on something. If you take a fatty acid off, it's called something else. But if you put a fatty acid onto something, it's called an acyl transferase that does that. So lysophospholipid will be re-acylated to form phospholipids. So that's going to be using the acyl transferases as well. And then cholesterol is also going to be added another fatty acid. So when I say esterified, this is just another way of saying you put a fatty acid on it. So when you esterify cholesterol, you put a fatty acid on it. And again, that's going to be using acyl transferases.
Dr. Fabi?
Yes.
So just to clarify, the long-chain fatty acids get turned into triglycerides by the acetyltransferase and then get transported by the chylomyricon and then get absorbed into the lymphatic system?
Yes.
Okay.
Which I will be repeating that in a second here, because we will talk about that as we move forward. But yeah, this is prior to making the chylomicron, this is what's happening. So as soon as they're absorbed, you have to remake the triglycerides, remake the phospholipids, and remake the cholesterol esters. So all that work your body did digesting them to absorb them in the intestinal cell, you just remake them. Remake the triglycerides, remake the phospholipids, remake the cholesterol esters, and then you're going to incorporate them into chylomicrons, and those chylomicrons will be put into the lymphatic system. And that is going to be in another slide, but I want to say this right here is important first. I need to mention this. Short chain and medium chain fatty acids are treated differently, as I've said a few times. They are not converted into triglycerides. They're going to be released into portal circulation, so they're going to go into the blood, and they're going to be carried by albumin. This was another commonly missed question from last term. Short chain and medium chain fatty acids are not carried by lipoproteins. They are carried by albumin. Albumin is a protein in the blood. A major carrier protein in the blood is albumin. It carries a lot of things. One of those things are fatty acids. It can carry long chain fatty acids as well. We'll talk about that later. But definitely after digestion, you do not need to incorporate short or long or medium chain fatty acids into chylomicrons. You simply put them in the blood, they bind to an albumin, and albumin will carry them in the blood. They're still lipids, so they still need protection from the water, but albumin can be their carrier. They don't have to be incorporated into lipoproteins. So that's what makes short and medium chain, one of the reasons short and medium chain fatty acids are different from these long chain fatty acids. Long chain fatty acids are going to require lipoproteins for transport, except for an occasion we'll talk about later. And so people who have lipid malabsorption disorders, we often will supplement their food with medium and short chain, because if they can't absorb or make chylomicrons properly, then you can just give them the short and medium chain and they should be okay. This is a really old drawing I made before PowerPoint had all these really good fun doodads. I was drawing things from scratch. But here we go. Short chain and medium chain fatty acids can go straight. This is the brush border, by the way. This is what I meant by brush border of your enterocytes. It's kind of, they create these microvilli. And short chain fatty acids can be absorbed, diffuse into the body, go straight to portal circulation, and go to the liver. Short and medium chain. But if it's the longer chain fatty acids, monoacylglycerols, cholesterol, phospholipids, or lysophospholipid, These guys go into the interior. So this is one cell, by the way. This whole thing is just a cell, one cell. You absorb all of these great lipids, and now these lipids go into the endoplasmic reticulum. The smooth endoplasmic reticulum can actually synthesize lipids. And so the smooth endoplasmic reticulum will remake triglycerides, remake the cholesterol esters and phospholipoproteins, put them into chylomicrons, which is a lipoprotein, and put the lipids out first into the lymphatic system. They don't go into the blood, they go into the lymph. They travel in the lymph through the body a little ways, and then they're put out into the circulation later. So, what is it, the thoracic duct of the subclavian blood vessel? My physiology there is a little, yeah, I think it's the subclavian vein, and that the lymph will drain into the circulatory system. So the chylomicrons will eventually end up in the blood, and then they'll travel. The job of the chylomicrons have one function, and it is to deliver all of those triglycerides that you just ate to the tissues, mainly your muscles and your adipocytes. Yeah. Endoplasmic reticulum. So there's a smooth and rough endoplasmic reticulum, so technically this is going to happen in the smooth endoplasmic reticulum.
So the cholesterol makes the chylomicron, or that's going into the chylomicron?
Cholesterol is going into the chylomicron. So the intestinal cell is making the chylomicron. The chylomicron is a lipoprotein, that looks like this. So it's kind of got, it's like a phospholipid membrane. And then inside here is all this great lipids that are going to be transported in this lipoprotein. So this is the chylomicron. So you notice that cholesterol is in here, triglycerides are in here. There might be other stuff in here like vitamin E. Your fat-soluble vitamins will also be in here. So vitamin A, vitamin E, vitamin K, what's the other one? D, vitamin D. So all of those will end up in there as well because they're lipid. And they're quite large, so they'll need to be transported using the lipoproteins as well. And so the chylomicron, again, goes into the lymph first. It ends up in the blood, and the one job of the chylomicrons is to deliver triglycerides to tissues from the intestinal tract. So it carries digested dietary lipids from the intestinal cell to the tissues to deliver triglycerides. What your muscles are going to do with triglycerides is use them for energy. So your muscle will take some of those triglycerides to use for energy, to make ATP. Your adipocytes, your fat cells, they're going to take those triglycerides and store them. So that's what's going to happen to the triglycerides that come from your food. They're going to go into chylomicrons, and they're going to be transported to your tissues, delivered to your tissues, and then eventually you're going to end up with what's called a chylomicron remnant, which is going to go to the liver. And the liver will figure out what to do with the chylomicron remnant at that point. And I don't want to get into that yet because we're going to spend a lot of time on lipoproteins. I think it might be next week, actually, or the third week. So chylomicrons transported will be eventually enter the blood, and so they're going to transport from the blood to the, in the blood to the tissues. It's going to be hydrolyzed at certain tissues. What are the two main tissues that are going to hydrolyze or break down the triglyceride, the chylomicron, to pull out the triglycerides? I already told you the two main tissues. Muscle and adipocytes. Muscle and adipocytes, they're going to be the two main tissues that are going to want to pull out those triglycerides and take them in. And that's called hydrolysis, so you can hydrolyze them. And the enzyme that's going to do that is another lipase. Lipoprotein lipase is going to be able to pull the triglycerides, the fat. They're actually going to take the triglycerides, pull the fatty acids off of them, and bring them into the muscle cell or the adipocyte cell. So again, it's another lipase, and like I told you before, lipases, they're the ones that break down triglycerides. And the reason the muscles and adipocytes are doing that is so that they can bring those fatty acids in to store them or to use them if the muscle, because the muscle can use it to make ATP. So lipoprotein lipase is associated with the blood vessel lining, endothelial. I had some physiology students that don't understand that term yet, so I'm going to introduce it now. So when I see you guys in quarter 4 in physiology lab, you already know what this means. But endothelial lining is just the lining of your blood vessels. So the endothelium, when you hear that word, it's just the lining of your blood vessels is the endothelium. And this lipoprotein lipase is not in any cell. It's actually just hanging out on the endothelium of your blood vessel walls. So it's just hanging out in the blood, attached to the blood vessel walls. So it's attached to the endothelium. And so as the chylomicron is swimming by in the blood, the lipoprotein lipase can actually grab onto it and help pull out those triglycerides, break off the fatty acids, and absorb them into the adipocyte or the muscle cell. So it releases the fatty acids, which are absorbed by the adipocyte or the muscle. Those are the two main tissues that will take it in. Oops. So again, remember, this is the intestinal cell. This is from your textbook. It's a little better picture. Here's the brush border. So they're going to be absorbed, the fat-soluble vitamins, 2 monoacylglycerols, the fatty acids that have been broken off, the cholesterols, all of that's going to be incorporated in the chylomicron. But remember, you have to rebuild the triglycerides, the cholesterol esters, all of that has to be rebuilt, and then they're incorporated into the chylomicron. Assembly and secretion of chylomicrons by intestinal mucosal cells. So that's another word for intestinal cell, is intestinal mucosal cell, because there's actually a lot of mucus that your intestinal cells have to make as well. So sometimes we call them intestinal mucosal cells. And again, short and medium chain do not require incorporation into chylomicrons. They can enter the blood directly on their own. And attached to what? What are short and medium chain fatty acids? How do they travel in the blood? Attached to what? Albumin. You guys are going to so ace my test. Okay. Oh, steatorrhea. So this is fat maldigestion or malabsorption. Can be caused by a lot of things. If your body doesn't properly make bile, remember the liver makes the bile, the gallbladder stores the bile, but if there's some sort of issue with the gallbladder, if it's blocked, then you can't get the bile into the intestinal tract, which means you can't absorb fat. You can't absorb lipids if you don't make bile. Pancreas, if you don't make the pancreatic lipase or the other enzymes, then you can't break down those enzymes. So that can lead to maldigestion and malabsorption. Defective intestinal cells. So if they can't properly absorb, then that can lead to issues. So steatorrhea is just a common name for any kind of malabsorption disorder related to fat. And basically, if your stool, the feces, if it floats in the water, then that shows you that you didn't properly absorb the fat from your diet. And so that can lead to a lot of issues if you're not properly absorbing important fats, especially your essential fatty acids. Oh my gosh, am I done? I don't know why I got excited. I guess because it's nice outside, and so we're not going to start, obviously, on the second lecture. We're going to have to do that week 2. That happened last term as well. We'll have time to catch up, so I'm not worried about it. I have to make a new lecture for the next chapter anyway that we're going to talk about. But do keep in mind, if you there is the lecture review video, so feel free to look through those. I don't have the review slides yet, but I'm going to try to make them. But let me know if you have any questions. I hope you guys have, what is it?
So far, is the video cracked? Some of the videos still cracked, just not the slides.